And welcome everyone again to another presentation. I have had my arm twisted, and so I'm here again to discuss RAC and more importantly Bisantrene. For those that don't know me, my name is Mason Hill. I have a degree in nutrition, a master's of dietetics. I have published a paper in Alzheimer's disease. And in that time, I've taken a particular interest in biotechnology companies. I have published material online following RAC's lead compound, Bisantrene, and it really is just a passion project. And so a lot of the work that I'll be doing today is the culmination of a lot of hours of work. And it is essentially going to summarize the clinical history of Bisantrene to date, which I would argue that almost no one in Australia outside of RAC management truly have understood to the degree that I'm about to show. I apologize for the length of the video. I am trying to keep it short. So let's go. Obviously, first of all, you are an adult and you need to invest accordingly. This is my analysis. I understand that I have a background in science, but the work that has been done on this presentation, the connections that I have made, these are mine alone, and I encourage you to conduct your own research. It's I am not liable for your investment. I don't encourage you to make an investment based on this research, but I do encourage you to do your own reading and come to your own conclusions. I am fairly confident about the work that I have done to date and how much I understand the compound, so I may make forward-looking statements, but I obviously cannot be held liable for them. And once again, I encourage you to do your own reading. Now, I have no idea how Rothman came up with 45 or so clinical trials. Bisantrene has been extensively used. There's, by my count, 65 clinical trials using the drug, the majority of which are in phase 2 trials. And of the patients that have been investigated, the majority are actually solid tumor patients, which is really quite interesting because the majority of the efficacy that we have seen for Bisantrene is in hematological cancer. Now, if you are unfamiliar with the story of Bisantrene, it is a compound that obviously has been used extensively in the clinic. It reached approval in France in the 1990s because the efficacy in acute myeloid leukemia was so good. For reasons that I will not go into in this video, the drug was lost due to a series of mergers and some technical things with providing the drug to patients, which have been sold. And so we're here today bringing the drug back to market. Something that I will be talking about a lot in this presentation is essentially the drug was never really used correctly. And there's a number of reasons that I could draw on to come to that conclusion. But essentially, if we look here, obviously the majority of the patients were solid tumor patients, and we have a disease control rate of 21%. Now, this is complete, partial, and stable disease, and the majority is made up from stable disease. And I mean, you can see the difference between hematological and solid tumor cancers if you compare the complete response rates and you compare the stable disease rates. Now, one of the major themes that we're going to be exploring in this presentation, and something that I've discovered that I feel is of utmost significance when discussing Bisantrene's clinical history, is how the drug was used. So, if you look below, the majority of the time, Bisantrene was used in a particular way for hematological cancers, and it was used in a very, very different way for solid tumors. Now, for those that are familiar with the RAC story and Bisantrene, you will know that previously researchers had thought that the drug was an anthracycline, so they dosed the drug in that fashion. Now, recent research has come out indicating that it is not that. And so how you use this drug in patients is very, very important because of the specific mechanisms of action that this drug has to drive an outcome. And so you can see down the bottom here, total dose per month is something that I have created because it strongly predicts overall response rate in this patient population. You can see a fairly significant difference here between the total dose per month for hematological cancers and solid tumors. So the discoveries, the rebirth of a first-in-class cardioprotective anti-cancer synergy drug. So Bisantrene is a very, very interesting molecule. So we have all of this clinical history, and around 2020, RAC had been going for a little while there. We have this discovery that amongst 260,000 molecules, Bisantrene is seen to be the number one inhibitor of this new protein, this new driver of cancer. And so it was really at this point where, when we look back, this is kind of the death of that anthracycline, that anthracene model. And when you take off the anthracycline glasses and you throw on this new FTO inhibition lens, it is essential to do that, but also to look back through the clinical history with that lens on. If you don't take or don't change the lens and you start to review the clinical history, I think that you will miss some of the significance, and that is why we're here today. I have spent significant time going through the clinical history of the drug, and I have done my best to look at it through this FTO inhibitor lens, and I will present some of that information to you today. Now, FTO inhibition is extremely important. It is a new developing field of therapy for cancer. It leverages on the M6A mRNA epitranscriptomic pathways. Essentially, it is the most abundant, the most frequent internal, so intracellular modification that controls the transcription, so the creation of genes and proteins that control cellular function. What that basically does is it controls the hallmarks of cancer, which are the characteristics, how cancer behaves. That makes it difficult to treat, that makes it spread around the body. And consistently we are seeing these M6A regulators, and particularly for what we're talking about today, FTO, being connected to these different hallmarks, these different characteristics of cancer. And so with a new developing field, we have a drug that, with all this extensive clinical history, it is now the most potent inhibitor of a biological target that has no commercial competitors. And in fact, it's so new that there are no clinical competitors. You have an untapped area of therapy that is connected to all the various different hallmarks of cancer, the characteristics of cancer. And you have a very strong clinical history where the drug has been approved previously in France. So it is a very exciting anti-cancer opportunity. Now in 2021, for those that have been following, there's also been a cardioprotection discovery. Now, this is very interesting because historically Bisantrene was created off the back of anthracyclines, as I said, and it was treated as an anthracycline. And the lead molecule, Doxorubicin, which eventually we're going to get a little bit more familiar with that drug. Doxorubicin was excellent at treating cancer. And it still is, it's widely used, but it decimates the heart. It basically turns it to ash. Researchers spent a lot of time trying to find compounds that had a very good anti-cancer effect whilst also being non-cardiotoxic. Now, fast forward 40, 50 years, and we find that Bisantrene, not only is it not cardiotoxic, it is in fact cardioprotective. This is the first time ever it has happened in cardio-oncology, ever. Millions and millions of compounds created over the last 6-7 decades to try and combat cancer, and there has never been a molecule that could effectively kill cancer whilst also providing cardioprotection. And so this is a very, very interesting opportunity, and it is indeed a rebirth of a first-in-class cardioprotection anti-cancer synergy drug. So FTO, why is it important? So I spoke before about the hallmarks of cancer, and it is very interesting. And what we're actually seeing is that FTO is a driver of a very wide range of cancer types. So across now 200 publications, FTO has been, or FTO overexpression, or decreased methylation states, is a primary driver of up to 32 different cancer types. This makes sense because the M6A mRNA mechanism is the most abundant internal modification within cells. Having a central network that controls a very wide range of functions makes a lot of sense. There is a growing body of research that suggests that RNA was here before DNA, long before it. And so these regulators of RNA transcription, are thought to be some of the most conserved mechanisms that we have controlling cellular function. It is no surprise to me that we see so many different cancer types with the overexpression of FTO as a key driver, because it has this central mechanism of controlling such a wide range of effects. Now, the cancer types that are bolded and colored orange are the cancer types where Bisantrene has demonstrated a response. So there are 22 different cancer types where Bisantrene has been shown to be effective. Both FTO and Bisantrene have a very wide range of cancer types where it's been shown to have an effect. So we have to keep in mind, this is an extremely difficult area to explore. It's extremely difficult to understand, and I've spent the better part of 4 years really aggressively researching this area. And I apologize if some of it goes over your head and I don't quite explain it in a way that you can fully comprehend the significance, but I will do my best. This figure, big shout out to Josh for generating some of the figures that are in this presentation. I could not have done it without you, my friend. You are a Jedi. So this figure is to highlight one of the most exciting things that I find about targeting the FTO protein. Now, these M6A regulators, they're fairly new to science, and they're fairly new to cancer therapy. Anyone who's invested in biotechnology companies, in particular anti-cancer companies, they're fairly familiar with the term of chemotherapy resistance or chemotherapy relapse. What it basically means is that it's a way in which the cancer cell resists death, and it can do it through a whole range of different methods, but essentially the cancer becomes very hard to treat. Now, the history of Bisantrene, it was deemed as this very effective salvage therapy drug where it just seemed to work when other drugs didn't. And so when you read that through the literature and then you become familiar with FTO at the core of chemotherapy resistance and chemotherapy relapse, even cancerous stem cells have overexpressed FTO, and that is a key driver for their unlimited self-renewal and the difficulties in actually treating them. If we look at this figure as FTO being at the very core, keeping in mind that all cancers are different and you're going to have some cancers that are not driven through this pathway. So if we have FTO at the core, this first ring is essentially the regulatory pathways. You could think about them as genes or proteins, transcription factors, signaling pathways, but they are the pathway FTO takes to elicit a certain characteristic of that cell, a certain function of that cell. And so the next band is our cellular behavior. It's how the cancer cell functions. I hope I'm not losing you. So through each of the modifications to these genes and proteins, the amount, you then control cellular function globally. Now, I like this figure because you have on the outer ring, you have some of the associated therapies. Now, each of these therapies has been shown in some way to synergize with FTO inhibitors or FTO knockdown models. I will just show you a groovy little table. So this is pretty much the information that I have on that today. And so what you'll see is these are drug classes on the left, and this is the relationship between an FTO inhibitor or an FTO knockdown model and each of these drug classes or drugs within these drug classes. And you'll see that there's actually quite a number. So I think there's now 26 different drug classes and 36 different individual drugs. And so when we come back to our figure, the approved drug classes can they fit the narrative of an outside-in approach. And I spoke before about FTO being at the core of cancer resistance. And so with an outside-in approach, these drugs are targeting proteins or transcription factors or pathways that lead to cell death. Now, each of these drugs on the outside here are targeting a specific component within where it might resist cell death or it might downregulate cellular energetics. And so this outside-in approach has been very effective in treating cancer, but you have a wide range of cancers that are resistant. You need to understand that this is not 100% accurate. Biology is extremely difficult. And it's like 100 pinball machines going at once, probably more. And so this figure is not entirely accurate, but I think it helps explain to some degree why FTO is at the core of resistance. Let's take PARP inhibitors, right? So they have 1, 2, and then I thought there was another one. Yeah, 3. So they have 3 different pathways in which they are targeting to drive cellular death, cancerous cell death. 3 pathways is good. You're targeting from various different angles, and it's going to be effective, right? But because FTO may be overexpressed, keeping in mind that this is an oversimplified example, but because FTO is overexpressed, it could be upregulating other pathways where this drug does not target, which leads to survival or resistance. The drug doesn't work. And so when we're reviewing the clinical history of Bisantrene and we see, it's really, really effective at this salvage therapy, it's able to work when other drugs can't, and it's sensitive when other drugs aren't. And so by targeting the center, we are using an inside-out approach, which has never been done before. Hopefully you understand my excitement now about being the most potent inhibitor of FTO. So, completely untapped area. There are no clinical competitors, there are no commercial competitors. The competitors that are within preclinical models at the moment, they're not very potent. And it's at this point, it's unsure whether they'll actually make it to being a usable drug. Now, a good example to help explain this figure, imagining that the cancer cell is a city, right? And at the core is FTO. It is a power plant, and it uses M6A to produce energy that drives each of these different hallmarks of cancer. Now, the hallmarks of cancer are certain organizations within the city. And I'll give you a couple of examples. Evading growth suppressors is, for example, upregulating the maintenance crews within the city and also the construction crews. You increase the hours at which they are working, so the growth never stops. Resisting cell death could be increasing the amount of hospitals that are being produced, so you're constantly repairing and replacing, and you're constantly at 100%. You could have scout and reconnaissance teams with activating invasion and metastases. So the power plant, the central network, is using M6A, it's removing M6A to then power each of these different hallmarks of the city of cancer. And so an FTO inhibitor coming in and blocking that process, you ablate all of that activity. Now, something that I want you to keep front of mind as I begin to go through some of the history, the clinical history of Bisantrene, is I want you to wonder what happens when you turn the power plant off for a day? What happens when you turn it off for 3 days? What happens when you turn it off for a week? And so if we use this example, turning off the power plant for 1 day in the month, the city might survive. If we turn the power plant off for a week, chaos might ensue. The next side of the coin is the cardioprotection discovery. It is a chemotherapy world first, as I said. The very interesting thing, and I have spoken about this before, is how FTO inhibitors influence heart cardiomyocyte viability. So if we look at this graph, this is essentially the number of cells that survive, or the percentage of cells, heart cells that survive. And when we provide cardiotoxic chemotherapy, there is a reduction in the number of survivors. When we also add in an FTO inhibitor or an FTO knockdown model, we see added killing. So more cardiomyocytes are dying, basically. The opposite is true for Bisantrene, where we see cardiotoxic chemotherapy killing cardiomyocytes. But when we add Bisantrene, we see this increase, so this blunting of effect. And so it's quite significant. In fact, it's on the right here, it is the same, if not better, than the currently approved compound Doxorubicin for preventing the cardiotoxicity associated with Doxorubicin, one of the most widely used anthracyclines there are. And so it's this protection, this ability to prevent cardiotoxicity from known cardiotoxic drugs that really sets Bisantrene apart from everybody else. It doesn't exist. It never has. And so the previous slide was the in vitro or in cell work. Now this is some of the in vivo or in animal model work. And I think it's at this point where I would like to clarify that it is impossible to compare preclinical studies with each other unless their study methodology is exactly the same, and every element of their study methodology. There are a whole number of ways in which study methodologies can be different. So I would like everyone that is listening and taking note to remind themselves that the comparisons that are being made between studies in preclinical models needs to be taken with a grain of salt. It is not a direct comparison. We're just looking for a similarity here. And so on that, this is capturing some of the in vivo work for both Doxorubicin and Bisantrene. Now on the left, we have some staining of fibrosis. It is a permanent damage to the heart where the cells have basically died. And so through our control here, we can see that there is virtually no damage. When you provide Doxorubicin to the animal model, you can see that the heart tissue has extensive fibrosis. You can see the difference between the two figures. Now, when you provide Doxorubicin and when you provide Bisantrene, you can see that there is virtually no change. The fibrosis is minimal. So if we look to the right, this is captured in a graph. So with Doxorubicin here, you can see that it's increased fibrosis by quite a large margin. And then by day 21 and day 42, Bisantrene has reduced that fibrosis, the same as Doxorubicin. So it is comparable. Looking below is a measure of left ventricle ejection fraction. We can see that obviously Doxorubicin decreases the left ventricular ejection fraction percentage, whereas both Bisantrene and Doxorubicin save it, and the same is true for body weight. So I'm a bit limited with the in vivo data. This is mainly because of the Cardioprotection journal publication that is going to come out in some time. I don't know when it's going to be, but I presume that a lot of the in vivo data for Bisantrene investigating it in combination with doxorubicin is going to be tied up in that. I'm very excited to see what that is. So the potential comparisons that could be made to dexrazoxane in the future hopefully are going to be a little bit more comprehensive. But what we're seeing here is both in vitro, in cells, in vivo, in animal models, we are seeing bisantrene demonstrating excellent comparison to dexrazoxane. One of the things that I saw, and I understand that you can't compare models, is the fibrosis studies used half as much doxorubicin in their models as the bisantrene models. So to be comparable both in vitro and in vivo to the only FDA approved cardioprotective drug is very promising. I think at this point you're probably wondering, well, why don't they just use dexrazoxane? And it's a little bit contentious. The dexrazoxane, if you look on the FDA label, you will find that the FDA label states that there is a risk that if you use dexrazoxane in combination with doxorubicin, there is a risk that you will reduce the efficacy of the treatment. Now I've gone through this and I've looked. There are some instances of that happening, and there are also instances of secondary malignancies. So the use of dexrazoxane in combination with doxorubicin has been significantly hampered through the risk of decreasing the efficacy of the anti-cancer treatment. This is not what we want. Cancer is a significantly higher risk of death than potential cardioprotection down the line. So this is why dexrazoxane really hasn't made it as a blockbuster drug, and highlights to some degree how unique bisantrene is to not only provide cardioprotection but have some established efficacy for killing cancer, which is what we're going to get into. We're entering into the territory of looking at both cardioprotection and anti-cancer synergy combined. It is difficult to comprehend this market purely because it does not exist. If we want to get a little bit crazy and think about what the ultimate cardioprotection anti-cancer synergy drug might be, there are a couple of things that are needed for the ultimate. First, you need broadness of action across a very wide range of cancer indications. You need to have strong anti-cancer synergy with a broad range of cardiotoxic chemotherapy. And then you also need to be able to protect the heart against the same chemotherapy. And if we think back to some previous slides, we can see that bisantrene, yes, indeed, is active in a broad range of cancer types. If we think to the FTO inhibitor and FTO knockdown models, also the function of FTO, we see that FTO inhibitors, FTO knockdown models are synergistic with a very broad number of therapies. Drug classes, there's about 26. I believe that there might be about 34 or 36 different individual drugs within those classes. And you have this FTO network that is really starting to be understood as the core of chemotherapy resistance and chemotherapy relapse. Bisantrene is able to protect against a very wide range of chemotherapies that cause cardiotoxicity. So from all of these different angles, we are starting to see that the ultimate cardioprotection anti-cancer synergy drug, bisantrene, is perfectly poised to leverage, to take advantage of that opportunity, which is excellent because there are no clinical and no commercial competitors that are able to effectively synergize with chemotherapy to more effectively kill cancer whilst protecting the heart from cardiotoxicity. Now, RAC have engaged the Oncolines cancer cell study set, and bisantrene has been shown to be broadly active at concentrations that can be achieved and have been achieved in humans. Anthracyclines themselves are very, very broad acting. So this is a major tick for us. The next thing is that Bisantrene has been shown to improve the cell killing of Doxorubicin in up to 86% of cancer cell lines. All of this preclinical work means that we have a drug with a strong clinical history of being safe and efficacious, reaching approval. You have cell studies that are showing that it's broadly active, which confirms the clinical history of the drug. You then have the same cell studies demonstrating that there is a synergy with this drug that is widely used in the market, but is very cardiotoxic. And you then have in vitro and in vivo models showing that the cardioprotection is competitive with the only FDA approved drug. Now, when I was trying to understand this, what does this actually mean? I looked at the Oncolines cancer cells and I was like, how representative? Like, it's cell studies. There are inherent risks. I mean, I understand we have the clinical history of Bisantrene, but looking at cell studies, there has to be certain limitations. And surely this does not translate. And then I started looking. The question that I suppose that I really wanted to answer here was, what is the likelihood chance that what we are seeing in vitro with Doxorubicin translates to humans? Now, the Oncolines cell library has been very extensively used in the literature, and there are many publications that anyone can go and read. But I suppose I was looking for very potent examples, and I really couldn't look past examples where the FDA had been involved. These are two different examples, one single agent and the other in combination. And what we see from these studies is that there is a very high level of agreement from the Oncolines cell library and what is shown on the FDA label for specific mutations that approved kinase inhibitors target. Now, where the Oncolines library are incorrect, or they did not correlate with what was on the FDA label, it is not that it was wrong. It was more so that a specific kinase inhibitor, it was not known that it could target mutations or those specific mutations. And so as a single agent, you have the Oncolines library correlating very closely with what is shown on the FDA label. The specific mutations that therapy is approved for in cancer aligns very, very closely to what they are sensitive in the Oncolines library. Now, that's great for Bisantrene as a single agent, and I mean, we know that Bisantrene has been widely active, 22 different cancer types. So there's a broadness of acting there, and we know that it can work. So as a single agent, seeing that 79% of the Oncolines cell library being sensitive to Bisantrene at clinically relevant doses, it's not really any shock. And I suppose what we would really like to be able to evaluate is the combination. Bisantrene has been shown in the Oncolines cell library to improve the cell killing of Doxorubicin by 86%. What is the likelihood chance that this plays out in patients? And this example is a doozy. So the publication was investigating the combination of dabrafenib and trametinib, and it found that this combination was specifically targeted to the BRAF mutation. Now, this work predates multiple FDA approvals, but one of the most hard-hitting ones is the one in 2022, where the combination was approved for virtually any type of solid tumor that expressed the specific mutation BRAF. It predated a blanket approval by the FDA 7 years. What I take from this is a very high level of confidence that the relationship that we see between Bisantrene and Doxorubicin in vitro is what we might see play out in humans. What we will discuss in time is, where are the goalposts for efficacy? And I think you might be fairly surprised that all we really need to see is a 15% improvement in efficacy. So for those of you who have been following me on social media, oh no, my figure is, this presentation is imperfect. My little figure's not orange. I've got flag IDA that's orange. This is an imposter. This is the real Bisantrene. For those of you who have been following me on social media and following the things that I post, you will know that Bisantrene is very effective in relapsed refractory AML. It was approved for that indication. Now, this had a particular dosing regimen that was used to drive that response. And so when I take that information and I compare it to other single agents, Bisantrene outperforms those single agents in complete response rates. So 50.7 versus the rest. And when I compare it to combinations, keeping in mind that this is the imposter, when I compare it to combinations, Bisantrene is very competitive in a very difficult to treat patient population. Bisantrene has very good efficacy when it is used in a particular dosing regimen. And this is something that we're going to go into in depth. And so in order to try and understand the response rates at a deeper level, I decided to conduct a series of tests to examine what were the significant determinants of objective response rate. Now, this was because I understood that there were inherent differences between cancer types. So a hematological cancer, for example, is going to have certain characteristics that are a little bit different from, say, a solid tumor. Now, the tissue type where that cancer originates from is going to have, even between solid tumor cancers, is going to have certain differences. Now, there are publications out there that have investigated these differences. And some of the things that come up are this cell cycle score, this cancer cell cycle score, commonly known as proliferation rate. Knowing that there are differences between different cancer types of different tissue origins, knowing that Bisantrene has demonstrated efficacy really only in one cancer type, but also knowing and appreciating that the total dose per month is different for that cancer type compared to everything else. So what I aimed to do is I ran a multivariate analysis, which is essentially you take the dependent variable, which is objective response rate, how many patients responded to the drug, and then you test the independent variables, which are these certain characteristics that might explain why the response happened. And what I found was the only significant determinant for objective response rate was the total dose per month. There was no other element of each individual cancer that determined to a level of significance the responses that Bisantrene has demonstrated in patients. I investigated FTO status using gene expression data. I investigated MYC, a known oncogene that is present quite frequently in AML. I investigated the proliferation rate, CCS, but the only significant determining factor for objective response rate was total dose per month. Dose, the amount of dose that you gave, didn't really matter and it didn't really correlate. What I took from this, and what I think others can take from this, is that there is no specific, just say that cancer is extremely complex, and its complexity makes analyses like this very, very difficult to do, because a cancer's true FTO expression status within a certain patient that is included into a certain trial is probably not going to be reflected by what is on genomic expression data, but it might be close. So it is impossible to say whether the patients included into the trials for Bisantrene over its entire duration of being used in the clinic, that their specific cancers represent what is in this figure here. However, that being said, there really isn't a relationship there. So what this is saying is that the determinant of a response to Bisantrene is not related to a specific element of that cancer. It is instead related to the amount of drug that you give per month. I want to expand on this more so that I'm sure that everyone understands what this means. Because total dose per month is the only significant determinant of a response, I then adjusted the response rate by the total dose per month. So you take the response and you divide it by the total dose, and it gives you an adjusted objective response rate. Now, if you are essentially taking out the variable, the independent variable of total dose per month, and you are just now looking at, well, if it did not exist, is there a significant determining factor? And so the figure on the right demonstrates that, again, there are no significant determining factors even when you adjust the objective response rate for total dose per month. Keeping in mind the limitations of this method, it again suggests that the responses that are seen with Bisantrene in patients is not related to some specific characteristic of that cancer, and it is instead related specifically to how much of Bisantrene that you give per month. Therefore, I could come to the conclusion that if Bisantrene was to be used in a way similarly to how it is used in AML in a broad range of other cancers, you would expect, based on this information, that you would achieve a stronger response to the drug. There are multiple ways in which we could run this analysis, and there are multiple different factors that could be contributing to them, and they're probably all interrelated to explain why Bisantrene is functioning the way that it is. The reason that I did this analysis is you have drugs like Doxorubicin. Doxorubicin is extremely good in hematological cancers, and it is as such because hematological cancers have a very, very high proliferation rate. They rely on this constant turnover of cells, this constant renewal. And so the way that Doxorubicin functions, it's very good at ablating that. Now, for cells that rapidly divide, rapidly proliferate, the disruption of that mechanism is catastrophic for them. However, for solid tumors, you don't have anywhere near as great a proliferation rate, as great a dividing rate as hematological cancers. So Doxorubicin does not necessarily work as well in those cancer types. The evidence that is put forward here suggests that Bisantrene is not like Doxorubicin. It doesn't require high levels of proliferation rate that are common in hematological cancers. There doesn't appear to be a relationship between FTO status and response rate, nor is there for MYC. And so this critically opens up to better responses being seen in patients when larger total doses per month are used. Now, there might be something that you could ask, and it's, well, why was Bisantrene used in this way in the first place? I mean, if we were seeing excellent efficacy in AML, why didn't the historical researchers just go, let's just use a little bit more? What was the limiting factor? And it's a really interesting question, and I think it speaks to the history of Bisantrene, its prior identity, if you want to call it such a thing. And the researchers of old believed Bisantrene to be an anthracycline-like compound, functioning in the same way that, say, other anthracyclines like Doxorubicin functioned. And because bisantrene was lumped into this identity, it followed on from the dosing regimens that other anthracyclines were used. So once every 3 weeks, anthracyclines were considered to be quite effective. Bisantrene, however, is not an anthracycline, and the discovery in 2020 forces investors to go back through the data set and really start having a look. Now, because the identity of the drug changed, I believe it is critical to look back through and evaluate relationships like this in much higher detail. I believe it to be essential because the core of the drug was critically misunderstood. The mechanisms by which bisantrene functions was critically misunderstood, and I think that this data opens the door to understanding the mechanisms of action for bisantrene, the unique way in which the drug functions that is different to the drug class that it was lumped into. I think it is unfair to compare bisantrene directly against its anthracene competitors when we now know that the mechanisms of action for bisantrene are very, very different. And I wonder if analyses like this can heavily inform future clinical trials where bisantrene can be used. And so the next few slides aim to showcase that. So what we can see here is that I have taken the solid tumor and hematological cancer data and I have separated them. I'm getting hot in here talking about this bisantrene. So we see I've taken both solid and hematological cancer and I have separated them. So we can see that there's quite a large number of data available. There is data available for 1 to 3 doses per month and daily for 3 to 5 days for solid tumor. But for hematological cancers, we see that there is a little bit more, where the drug is used a little bit more frequently and at much higher doses. So evaluating the figure on the right shows that, where you see in this histogram, you see that solid dosing, total dose per month, is actually very, very small, whereas hematological is massive. So there is an enormous difference between the dosing that was used in solid cancers and hematological cancers. Now, it caused me to wonder. I was like, well, is there any examples where bisantrene has been used in a way like AML, but for solid tumors? I wondered what might happen. Unfortunately, when you look at the history, the 3 and 5 day daily dosing for solid tumors all used a very, very low dose of bisantrene. The results are right there for you to see. But if we look at the 2 most recent clinical trials, SHIBA 1 and SHIBA 2, there is something here that I want to be quite clear, as this is up to the individual to decide how they wish to take this information, how they wish to review this information, but this is exploratory for me. I don't necessarily take this as 100% fact, but I felt like it was significant enough and helped me to explain the data through a new lens. So when we look at the SHIBA 1 and SHIBA 2 trials, we know that we have a 40% response rate, very good for a heavily pretreated patient population. 10 patients responded. Now, 7 of those 10 had extramedullary AML. There were only 9 extramedullary AML patients that had been included into the 2 clinical trials, which gives an overall response rate of 77.8%. Now, extramedullary AML it's not necessarily very new, but its understanding and the role that it plays in AML is only really now starting to be understood. How frequently it presents, how do we best treat it? It's a difficult cancer to treat. But one of the major things that I found extremely interesting about extramedullary AML is how similar it is to solid tumors. And I've taken a quote here, and I'll read it for you. Striking histological similarities to solid cancers were found. A single filing architectural pattern virtually identical to that of invasive lobular breast carcinoma and dense desmoplastic keloid-like fibrosis similar to colon, gallbladder, and pancreas carcinomas. There are many publications out there that are starting to showcase the similarities between solid tumors and extramedullary AML, also known as myeloid sarcoma. Now, myeloid sarcoma is characterized by the World Health Organization as not a hematological cancer, but it's also not a solid cancer. It is a hematological cancer that has created a mass and has taken up tissue residence outside of the bone marrow. And it is a mass, but it is unlike either hematological or solid tumor. But it has characteristics. And so we have a patient population. I understand that it is 9. I understand that it's 9 patients. We're limited in what we can draw from this, but we have 9 patients included into the 2 most recent phase 2 trials, where 78% of them are responding, and the cancer types are solid tumor-like metastatic AML cancers. They have characteristics of solid tumors, and the dosing pattern that was used in these cancer types similar to what was used historically in AML that led it to be approved. When we take the extramedullary patients away from the hematological group and we place them into the solid cancer group, we see that the trend lines for each of them start to marry up, and they show a lot of similarities. I want to reiterate, this is exploratory. This isn't necessarily meant to be stated as fact. This may not eventuate. But what we see here, the relationship that we see between dose amount and frequency, total dose per month, is aligning more to the relationship that we see between total dose per month and objective response rate. This is it depends on, I want to make sure I say this correctly. It is up to each investor to determine whether the relationship that they see on the graph on the right might be what plays out to some degree if the drug was to be used in a particular way that increased the total dose per month. My opinion, I strongly suggest that this is an indication of what we might see when bisantrene is used in a way that leverages off its mechanisms of action. Talking specifically about increasing the total dose per month for bisantrene, I believe that we will see responses similar to this effect, just like we see in AML. And there are a whole host of reasons why I think that will happen. It is because of the inconsistencies. There are many of them where bisantrene is showing efficacy that outstrips the competition. These are approved drugs. These are drugs that work. They kill cancer. They're effective. They've been used for years. They save people's lives. And we see in vitro models where bisantrene is outperforming these drugs by no small margin. This figure on the bottom left, mitoxantrone was not used. Bisantrene is 3 times more sensitive than doxorubicin. 3 times. In all of these preclinical models, all of them, this is not a selection, this is all of them. Bisantrene outperforms approved therapies. It is one of the most sensitive drugs of that era. And so when you look at that, you go, okay, well that's very interesting. We have a drug that seems to be working quite well in vitro, and then we start putting it into patients. And I mean, if you remove the hematological cancer patients, you go, well, you remove the hematological patients, you go, this drug's shit. The objective response rate is terrible. When you add the hematological patients, the patients that we used were dosed in a particular fashion, you go, there's something to this. And so the inconsistencies is really the only thing is this dosing regimen. When you use the drug in a particular way, it aligns exactly with the in vitro models. So if we look specifically, get out of here. Relapse refractory AML, better than the competition. Extramedullary AML, better than the competition. In vitro, better than the competition. But for solid tumors, Bisantrene was not used in a way that leverages off its mechanisms of action. Now, let me show you some other interesting stuff. So, one of the most fascinating things that I find about this drug is the hematological data. And if you've been following on through the RAC story, or if you're just new to it now, you'll know that Bisantrene was approved for AML for outstanding efficacy. It's, as I've shown, it's better than some of the single agent drugs that are used for that indication. So you'd be fairly confident, as I am, to come to the conclusion that, well, the drug works. In AML, it works, and it's very effective. I mean, if you want to be conservative and say, oh, well, let's just say it's as good as the competition, no problem. The question that I have for you, you've been following on, I want you to think about, how will Bisantrene function? What will the efficacy be if you dose Bisantrene in the same way that they dosed it for solid tumors, once every 3 weeks? So hopefully you have had time to ponder that. And so we're going to come down here. So, this is the data set for Bisantrene. This is for all the little data nerds out there like me. This is it. So what I'm going to show you is when you dose Bisantrene like solid tumors. So we have here a phase 1/2A. It is labeled A because there is a phase 1/2B that was used where they used solid tumors. Now this is one large study where I've separated the patients, the patient populations into solid tumor and AML. This is done for me so that I could run stronger analyses between groups. And this phase 2 component took a certain number of patients and gave them a phase 2 dose. Now you'll see here that there is a once every 3 week dosing regimen. The dose range is between 190 and 430. The 190 milligrams per meter squared once every 3 weeks, that is the phase 1 component. The phase 2 component used 300 to 430 milligrams per meter squared once every 3 weeks. And so the 19 patients that were included into that phase 2 trial, you have one complete response for a grand total of 5% response rate. Bisantrene, previously approved in relapse refractory AML for outstanding efficacy, demonstrates a 5% response rate when it is used once every 3 weeks in the same indication. So when we look at, oh gosh, my little mouse thing. This is it down here. There is a 10-fold improvement in efficacy when you dose Bisantrene from a once every 3 weeks to an everyday for 7 days. Tenfold improvement. So when you go above and you see Bisantrene dosed once every 3 weeks and how that compares to a solid tumor like metastatic AML cancer, you see a very, very similar relationship between those two cancer types. Now, you might go, it's not quite enough to get me over the line. So I do have a little bit more. So when we come back to my wonderful spreadsheet and we look at, let's go up the top. So we've got, what is going to be better to see? Yeah, this is fine. We have 4 phase 2 trials where Bisantrene has been used once every 3 weeks. And so if we focus in on these 4, breast, colorectal, and lung cancer, I'll go a little bit closer. Yeah, that's better. And so we see that for these 4 trials, we have 20%, 20%, 0% response rate. But the disease control rate for these papers is between 50 and 70%. So in a solid tumor trial where Bisantrene was used once every 3 weeks, you have poor response rate, but you have up to 70% of the patient population responding in some way to the drug. Now, some of these responses, the stable disease, they actually lasted for quite a while. You have 26 weeks, 9 to 15 weeks. And so even though Bisantrene was not used effectively, the dosing regimen is not in alignment with anything that we have discussed here. You have a very large percentage of the patient population that are achieving a stable disease. Now, I liken this to an antibiotic example because I feel like it explains it. It explains it in a way that people understand it. I have the best antibiotic in the world, and I'm going to give you this antibiotic once every 3 weeks or once a month. Now, it doesn't matter how good that antibiotic is, it is not going to treat the bacterial infection, but it might show a response. When you give the antibacterial as it is intended every day for 2 weeks, then it is able to function in a way that aligns with its mechanisms of action. This is a similar type of phenomena that I feel is playing out with Bisantrene and explains the inconsistencies that we see in solid tumor-like cancers. Bisantrene is not effective once every 3 weeks, even in AML where it has achieved previous approval and it outperforms the competition as a single agent. It's not effective. Similarly, it is not effective in solid tumors where it is used once every 3 weeks. But in a solid tumor-like cancer, metastatic solid tumor-like cancer, you have excellent efficacy, better than the rest. Both of these align with the in vitro work where you have Bisantrene outperforming the competition. The only thing that doesn't make sense is the dosing regimen. And it's because the researchers that historically used the drug thought it was something else. We know it's different. We now know that the drug is targeting different mechanisms. What I would like to bring in now is the concept of multiple mechanisms. We're familiar with the targeting of FTO. It's the most recently discovered and it is most potent action for Bisantrene. But there has to be something to explain the difference in efficacy that we see even when a frequent dosing regimen is used. So when the drug is used, low dose over 5 days, I've excluded everything else, we see that the response rate is still fairly low. When you increase the dose, even on a 5-day schedule, you see an increase in response rate. And then once again, more dose per month, you see once again an increase in response rate. What is explaining this phenomena? What explains it is bisantrene is a pleiotropic functioning drug. It has multiple mechanisms of action. Now, this is in alignment with all of the drugs that were created in that era. Chemotherapy was pretty much a one drug mechanism. So if someone has cancer, we provide a single drug that targets a very wide system. A good example is Doxorubicin, that has a very wide range of targets. And so bisantrene is like those drugs, and if you look at the historic literature, there are actually quite a number of targets that have come up. Now, historically, it was thought to be an anthracycline and participate in DNA intercalation. Now, if you review the clinical history, the preclinical history for bisantrene, you'll see that doxorubicin intercalates into DNA 155 times stronger than bisantrene. So what are the other mechanisms that explain the efficacy that we see for bisantrene? And I've labeled them down here at the bottom. So this is like a stepwise ladder example. And so if we imagine that there is no orange liquid in there, and it begins to fill, the potent mechanisms, as this starts to fill, a greater percentage of these mechanisms will be in effect. The slightly less potent ones, they will have a percentage, but it will be less than the very potent mechanisms, but more than the less potent, and definitely more than DNA intercalation. And so once you've reached this low threshold, 100% of both FTO inhibition and A1 adrenoceptors will be in effect. And a percentage of the rest, in a low dose every day for 5 days regimen, we have less liquid coming in here, so a less grand total percentage of all of these mechanisms working in synergy. As we increase the dose every day, we have a greater percentage of each of these different mechanisms being able to contribute to the total cytotoxic action for bisantrene. And I think it's this mechanism that explains why we see this 5-fold improvement in objective response rate when we just increase the total dose per month. Bisantrene is a pleiotropic mechanistic drug, as is doxorubicin. Many mechanisms of action that are shared between, that are targeted by each drug. If doxorubicin is a DNA targeted shotgun, a pleiotropic shotgun, bisantrene is the RNA equivalent. So if doxorubicin is targeting DNA, bisantrene is targeting RNA. And each drug individually has its own targets, its own biological targets that contribute to its cell killing. And we really see that in some of the in vitro studies which have done comparisons to both doxorubicin and bisantrene that were done about 40 years ago. Now, the very interesting thing for me, which is in alignment with what I have been saying so far, is that bisantrene in these cell models, it is sensitive 26.5% of the time where doxorubicin is resistant. It is sensitive in cancers 26.5% of the time where doxorubicin is resistant. Now, conversely, doxorubicin is sensitive only 4.4% of the time where bisantrene is resistant. So clearly there is a patient population that is sensitive to this RNA targeted shotgun effect of bisantrene where doxorubicin is not. Now, each of the mechanisms of action for bisantrene has in its own way demonstrated to be synergistic with doxorubicin. I think that this is why we see such a large percentage of additive cell killing when you provide bisantrene in combination with doxorubicin. Each of the mechanisms in their own way are able to synergize with the effects of doxorubicin to improve in cell killing. So, the upcoming Cardioprotection anti-cancer synergy trial, where all of this craziness that I've been talking about gets tested. how dare you talk about all this science and make a logical point. The Cardioprotection Anti-Cancer Synergy trial. Oh boy, we have been waiting for this for a long time. I am very excited. If it is not easy for you to tell by the way that I speak about this opportunity, I am very excited for this. I think we are perfectly poised to see a benefit both through anti-cancer efficacy and cardioprotection, so much so that I am looking at the high end for both. I don't know if I'm quite prepared to publish what I think will happen online, but I can say that I expect the results to be significant. And this information is taken from the Triangle Report, where they believe that a high anti-cancer efficacy is a 15% improvement in complete response rate or a 4-month improvement in median progression-free survival. Now, as I said before, an effective cardioprotection anti-cancer synergy drug wants to be broad targeting. So I understand that this is in breast cancer or various types of breast cancer, but I would be looking across the board at all of the potential indications, seeing this anti-cancer efficacy. I want to see it all, and I think that we can get there. Now, the cardioprotection element, you want to see a 30% reduction in the incidence of cardiac events. Now, I've taken the Howden data where they used VO2 peak to determine the influence on anthracycline treatment on functional disability and cardiotoxicity. So they're about an 8.5% in VO2 peak. Now, this contributed to 8.5 years of regular aging. So through the treatment of anthracyclines, you had almost 10 years of your life taken as a percentage. That is a big percent. And so a 30% decrease is essentially 6% or 6 years. And because of what I see in vitro and in vivo for Bisantrene providing cardioprotection that is in alignment with dexrazoxane, and what I know of dexrazoxane clinically, I suspect that we may even see something better than this. Something more to the degree of, say, 60 to 70% reduction. If we can, through the phase 1A/B component, show a signal of cardioprotection and show a signal of anti-cancer efficacy in synergy with Doxorubicin, we will be perfectly poised for a blockbuster drug. Again, not financial advice, but it has never happened before. Not once ever in oncology have you been able to pair a drug, 2 toxic compounds together, kill the cancer better but protect the heart. It is so rare that it is difficult to try and help you understand the significance of how important a discovery like that is for patients around the world. And this takes us to one of the most interesting elements of Bisantrene's clinical history, the circumstantial cardioprotection anti-cancer synergy data. Now, Bisantrene wasn't always used as a single agent, and it has been used in combination with other drugs. We have 2 older studies in pediatric patients that Bisantrene was used in combination with etoposide, carboplatin, and cytarabine. Now we have a recent example of Bisantrene being used in adults in combination with clofarabine and fludarabine. In all cases, all 60 patients, they were all heavily pretreated with anthracyclines. From the 60 patients, 45 of which were children who are at an extremely heightened risk of cardiotoxicity. There is a 1.7% cardiac event rate across 60 patients heavily exposed to anthracyclines. We have seen through in vitro models that FTO knockdown models increase the cardiotoxicity of chemotherapy, cardiotoxic chemotherapy. if that translates into patients, we have a very interesting thing happening where, I mean, if we use this as an example, etoposide and carboplatin, toxic for the heart, kill the cancer. We have bisantrene through its FTO inhibition mechanism, adding to the cardiotoxicity going towards the heart, but also synergizing with these to more effectively kill the cancer. But then we have the cardioprotection mechanism that is preventing that damage done to the heart, keeping in mind that these patients are heavily pretreated with anthracyclines. Coming into the trial, their hearts are damaged and weakened. And we see a less than 2% cardiac event rate in this entire group of patients. It becomes very exciting. I decided to generate this table. I feel like it is of particular significance. Clearly, we do not have combination data where bisantrene has been used in combination with doxorubicin in humans. I generated this table with the different ways in which doxorubicin, or anthracyclines in general, lead to cardiotoxicity. And so the number one contributor from anthracyclines, and more specifically doxorubicin, is reactive oxygen species production, ROS production. And so from this, I was like, well, I wonder what the shared mechanisms are for cardiotoxicity for all of these drugs. So we have the drugs that we have shown cardioprotection in preclinical models and in humans. And we see something very interesting in that ROS production is consistently seen across every single drug class. Now, reactive oxygen species production influences inflammation. It influences cardiac remodeling, apoptosis and necrosis. And so we have a central mechanism which is pretty widely regarded as the primary mechanism for cardiotoxicity for doxorubicin. And we have bisantrene demonstrating cardioprotection against these two drug classes in vitro, and these three drug classes in patients, all of which were heavily pretreated with anthracyclines, and 75% of which were children, the most sensitive to cardiotoxic drugs. Dexrazoxane targets mostly the disruption of iron homeostasis. It is down here on the list. It also influences topoisomerase 2 inhibition. I want to be very careful about what I say. It seems like I'm doing that a bit. We have potentially, now I'm going to tell you what I think the cardioprotection mechanism of action is in a little bit, but we have the potential for a cardioprotection mechanism that is universal. Means that any cardiotoxic drug where it influences cardiac events, long-term cardiovascular function, we have a drug that can protect against it, no matter what. I think and I've done a fair bit of reading here, I think the cardioprotection mechanism is not a chance. We're gonna have to wait for that one. I think I know what it is, but no, I'm not telling any of you. Just briefly, I mean, I'm not going to go into too much detail. This list here captures some of the cardiotoxic drugs that have been shown to synergize with FTO inhibitors and FTO knockdown models. So if you have an RNA targeted shotgun like bisantrene that has been shown to be effective across a wide range of cancer types, it is broad acting, but it also has universal cardioprotection. You have a very interesting drug. Broad acting anti-cancer effects, broad acting synergy. The mechanism has been widely shown to be synergistic. and you have universal cardioprotection. It's a very interesting drug. I understand that we are exploring Bisantrene in combination with Doxorubicin, but it opens up the potential for many other combinations where cardiotoxicity is frequent, where synergy and added anti-cancer effects would also be required. Because there are no drugs that do this, it is a blue water opportunity. It is untapped. So there is circumstantial clinical data that suggests Bisantrene is and has been cardioprotective. All of the drugs that Bisantrene has been cardioprotective against have a similar mechanism of cardiotoxicity. That similar mechanism of cardiotoxicity is the primary mechanism of cardiotoxicity for Doxorubicin. The availability of all of this data suggests to me that I am extremely confident that we are going to see a positive result and a world first in the upcoming phase 1A/B clinical trial. All we need is a signal to be a world first, the first ever. I am very, very confident, possibly as confident as you could be going into a clinical trial where, anything can happen.
I'm not sure.
We've got Siri talking to me over there. Lastly, this figure is, I think, one of the most interesting things that Josh has created for me, and it depicts the path that millions of people have taken. Millions of people have taken recently this year, and millions of people will continue to take into the future. And Bisantrene represents an option. And so if I could talk briefly about the path that people follow. A person, let's say it might be you or it might be a loved one, is sitting across from an oncologist. You may have failed a previous line of therapy, or it could be your first. But in any case, you are in consultation with this oncologist, and they say to you, look, we have this drug, we've used it for years, it's really effective, but the thing is we're kind of limited on how much we can use. It's really like it's a balancing act. If we use too much, it might damage your heart for many years to come, but it'll most likely treat your cancer, so it's really effective. If it's working, we might have to give a little bit more than what we have to. And for millions of people, they are essentially forced into having to make a decision where I'm prepared to sacrifice my heart to kill the cancer so I can, live the rest of my life. And so up to the point of a successful treatment, you have cancer threat increasing and quality of life decreasing. And then once you've successfully treated the cancer, all of a sudden quality of life snaps back up. Like, yes, like I've done it. Like it's over. Now there might still be a lingering feeling of cancer, like, is it really gone? Is this it? And what you see is as that cancer threat begins to dissipate, you have this increasing looming threat of cardiovascular disease. Now, it might not necessarily be as in your face as I'm going to have a heart attack, but it could be like, I'm struggling to get up the stairs. I can't run like I used to. I can't play basketball like I used to. Depending on what age your diagnosis is, if you are a child, you are significantly more likely to develop some sort of cardiovascular disease than aged matched controls. If you are an adult of any sort of reasonable age, you are significantly higher risk of having a cardiovascular disease than aged matched controls. And so as you start to move through your life, you are heavily impacted by your cancer treatment because your heart has been permanently damaged. It will never improve. So you may never have cancer again, but for the rest of your life, your quality of life will continue to decrease as the effects of anti-cancer treatment are felt. This is the pathway for millions of people, and it has been for decades. decades. Bisantrene, if successful, provides a different discourse to those same patients. Sitting across from the oncologist, either you or your family, if you were presented with that option, and then he also said, but we have this drug, it has a greater percentage chance of killing the cancer, as well as it will prevent the cardiotoxicity to a certain percent that you would have experienced from Doxorubicin. Do you want to spend the money to save your heart whilst also more effectively killing the cancer? Most of the time. If it was me and I had to make a decision for myself and for my quality of life, it would be a no-brainer. If I couldn't move and function and live long into my years, it would be devastating for me. And so having that option available to you, knowing that because healthy heart tissue is more predictable than cancerous tissue, you could be more confident that cardioprotective effects of Bisantrene are going to work for you. So at the very least, you are protecting yourself from lifelong permanent heart damage. So the pathway for that person looks very, very, very different to the pathway to the person that does not take Bisantrene in combination with Doxorubicin. Admittedly, this is if it is successful. So instead, you have a significantly reduced cancer threat following a successful treatment. Because I took a combination, it works better. The cardiovascular disease threat, I've taken something to protect my heart. So the threat is not necessarily as there. And quality of life, it stays elevated for longer. You're able to enjoy more of your life for longer. Now, in terms of quality of life, we have a thing called disability adjusted life years. Now, it is a measure of the morbidity of disease, how much of the quality of life it removes from a person within a population. And so at the very, very top, we have cardiovascular disease. But second, we have cancer. And Bisantrene essentially fits into two of the most influential non-communicable diseases that there are and solves a major problem in oncology. Never has an option been available to millions of patients each year to prevent the cardiotoxicity of an effective anti-cancer treatment whilst also making that treatment work better. If you were sitting across from the oncologist, what decision would you come to? If it was me, there would be no question. This, I feel, is where Bisantrene fits into oncology, and it is why I think it will be a successful drug if cardioprotection anti-cancer synergy is proved in the clinic. And that pretty much wraps it up for me. So thank you very much for listening. If you made it this far, like an hour and a half. If you made it this far, well done you. I'm impressed, I'm proud. I'll keep this pretty short. I just thank everyone who listens, who reads my stuff and who comments. It means a lot. I thank a lot of the people who actually contribute and provide excellent discourse. I don't really need to say too much more about Bisantrene or RAC. I think the sooner that we can get it into the clinic, the sooner that we can evaluate its effects, the better. I will provide a reference list for all the papers that I cite throughout my work. And thank you very much for listening. I might see you on the next one if I'm roped into it again. And thank you very much. Take care.